moving into Chapter 15, Mechanisms of Pathogenicity, we are going to spend some time learning some different terminology, terms that you may have used in the past, but maybe not used exactly correct. Maybe we'll learn some new terms. I never like to tell a group of students that they need to memorize, but you do need to spend time learning the different terms, the definition of terms, so that you can then go further and make sure that you can use those terms properly. So jumping right in, let's first look at different characteristics of infectious diseases. If we're going to spend a chapter discussing diseases, we need to make sure we understand what the difference between a disease and an infection is. A disease is any condition in which the normal structure or functions of the body are damaged or impaired. A disease can be caused by an infection, but there are other causes of disease. There are genetic diseases. There are, accidents that can lead to different diseases. What we're particularly interested in this class are infectious diseases. So, in order to have an infectious disease, you need an infection, which is successful colonization of a host by a microbe. Now, not all infections can lead to disease. We have infections of our normal microbiota. They don't cause disease. But an infectious disease is a disease where an infection of pathogens has led to this change in the normal structure or functions of the body, causing disease. And we've been using the term pathogen, but just to be complete, a pathogen is a microbe that causes disease. So in your past, I know you've used the term signs and symptoms of a disease, but have you used those terms correctly? A sign is any objective, measurable, or observable result of a disease. So these are things you can actually see or measure. So if I have a high temperature, my heart rate has increased, my breathing rate has increased, my blood pressure has gone up, gone down. Those are all things you can see. Those would be signs of a disease. Symptoms are much more subjective. These are things that may be important, but they're not something that we can measure or see. Only the patient can experience. Things like nausea, loss of appetite, pain. Now, I hate to say this, but I always could keep it straight when I was trying to determine whether or not something was a sign or a symptom. If it can be faked, it's typically a symptom. Because I can stand in front of you and tell you that I'm nauseous or nauseated or that my arm's hurting. You have no way of knowing whether that's true or not. But if I tell you I have a high temperature, you can measure that with a thermometer. Every disease that we'll discuss throughout this semester has a syndrome. The syndrome is the group of signs and symptoms that always occur with a particular disease. Most of the time, you are going to rely more on the signs of a disease than the symptoms because they are measurable. So now let's look at a few different ways that we can classify diseases. So we already talked about an infectious disease. So let's look at communicable, non-communicable, and contagious. A communicable disease is a disease that can be spread from one person to another through many different mechanisms, directly coming in contact with the person or even indirect contact. But communicable means one person has it and can give it to someone else. So a great example of that would be something like gonorrhea. That's sexually transmitted. So in order to transmit that disease, one person has to touch the other. So it's a communicable. You only get it from someone else. You can't get gonorrhea from the dirt or drinking contaminated water or anything like that. Non-communicable means that you can't get that disease from another person. You're going to get it from something like a water source, a food source, soil. The example I have on the computer is tetanus. Tetanus you can only get from a spore in the dirt going into your body. So you can be around somebody that has tetanus forever, and you're never going to get tetanus from that person. A contagious disease is a communicable disease that is spread very, very rapidly, very easily. So, my example here is measles. Measles is spread by simply breathing the air that someone who has measles has breathed. So it's pretty easily spread. Something that wouldn't be highly contagious would be a disease that you'd have to be around somebody for a long period of time before you would get it. So something that comes to mind would be something like leprosy. You could live with someone for 10 years and never contract leprosy. You might, but it's not highly contagious. A nosocomial infection is a disease acquired while you're in the hospital. In order for it to be classified as nosocomial, it must not be the reason that sent you to the hospital. So it has to be a disease that's caused by something different than what you went into the hospital for. A zoonotic disease is a disease spread from animals to humans. So zoonotic is a type of non-communicable disease. An example would be rabies. To get rabies, you must come in contact with an animal that has the disease. When we think of diseases, they all exist in 5 specific periods. Every disease follows this pattern. There's always step 1, incubation, where the pathogen is multiplying in the host. So on my graph, it's going to be in this region. You see you have very small amount of pathogen because they're trying to get established. They're trying to replicate within you. During the incubation period, you will have no signs or symptoms. You have no idea that a disease is coming. In the prodromal period, our next phase, the pathogen keeps on growing, and this is where you kind of start to feel bad. You get some signs and symptoms, you know, uh-oh, I'm getting sick. The next stage, the period of illness, this is when you reach your peak amount of pathogens in the body, and this is when you feel the worst during your period of illness. Period of decline begins once the number of pathogens starts to decrease. Your signs and symptoms will still be there, but they'll start getting better. In the period of convalescence, the patient is going to return to normal function and the number of pathogens in the body will disappear. One thing that most people are not aware of is if we're discussing a contagious disease, it is actually contagious during all 5 periods. Even when you're in the incubation and period of convalescence and you may have none or very little signs or symptoms present. So just a couple other ways we can classify diseases. Diseases can be considered acute, which means that they're occurring just within a short period of time, hours to days, and then you get better, like the flu. A chronic disease is a disease that causes changes in the body that spans a long period of time. Months, years, even a lifetime. A latent disease occurs when a pathogen makes you sick and then it stays inside of you lying dormant for different periods of time, and then it comes back out actively replicating, making you sick again. So an example of latent would be chickenpox coming back as shingles, or something like the herpes virus. If we think about fever blisters, you may get a fever blister today and then it goes away in a couple days. And you may not get another fever blister for 6 months, for 2 years. All that time when you didn't have the fever blister, the pathogen was living inside of your body. We also need to consider the terms primary pathogen and opportunistic pathogen. Whenever we get sick, there's always a pathogen that has caused disease. That's your primary pathogen. There are sometimes opportunistic pathogens that are only going to cause disease when our immune system is busy. So you can think of, we may already have a primary pathogen infection. It's got our immune system busy. So then that little opportunistic pathogen says, oh, okay, I'm going to start replicating too, and maybe I can make them sick. There are 4 stages of pathogenesis. If we think about, so So this is different than the stages of the disease, the infection. This is thinking about the pathogen actually, what does it do to your body? First, you're exposed to the pathogen, and the pathogen has to get inside of you. So remember those portals of entry, like the parenteral route. After exposure, then we get adhesion. The pathogen may get into your blood, for example, but if it's going to infect your liver, it's got to be able to attach to specifically your liver cells. Invasion is third step, and that's when the pathogen goes throughout those local tissues and parts of the body that it wants to infect. And then the last step is infection. There's different types of infection. An infection can stay local, which means it's localized to one specific region, one pathogen. A systemic infection occurs when the pathogen spreads throughout your body, typically through the bloodstream. A focal infection can occur whenever you have a pathogen leaves its first site of infection, spreads through the body, and then starts a secondary location of infection. This is really common with a lot of our bacterial infections of the heart. They may start out in other places, travel through the blood, and then start a new infection location in the heart. The primary infection is the infection with one pathogen, and then secondary infection is when another pathogen comes along because the body is weakened. And just to be complete, since we looked here at these portals of entry and said that's step one of pathogenesis, we have to have basically exposure to the pathogen. To close out this cycle, the pathogen is going to leave through one of the portals of exit. The last topic in this chapter that I wanted to discuss with you is one type of virulence factor called toxins. A virulence factor is something that makes the pathogen really capable of making you sick. And so there's several different majors that a lot of you will go into after this where you need to be able to distinguish between an endotoxin and an exotoxin. By definition, the endotoxin is within the outside membrane of the gram-negative bacteria. So remember, when we talked about gram-negative bacteria, we learned that gram-negatives have an extra lipopolysaccharide layer outside of their peptidoglycan cell wall. Pieces of that are the endotoxin. So since we're discussing something that's part of the cell, for the endotoxin to release, the cell has to die. So it's kind of like a ha-ha, you killed me, but now deal with my endotoxin. The exotoxin is capable of being produced while the bacteria is still alive. So the living cell itself will actually spit out that exotoxin. So I found this table that I thought may help you with this a little, and so I've copied that from a different source, just so you'll have a little more information on the differences between endotoxins and exotoxins.